The first stage of mitosis, and we'll have 4 stages, and we'll be naming them according to what's happening in the stage as opposed to its actual scientific name. But the first stage is called the condensing stage or phase. Here we see 2 cells. Again, these are plant cells. Here's one over here, and here the nucleus is evenly stained, so we're not seeing chromosomes. And whenever you see the nucleus evenly stained like this, you are not in one of the stages of mitosis. You are in the interphase. So you can tell that a cell is in interphase when the nucleus is evenly stained. Here, though, this cell here, here's the wall of the cell, here's the nucleus. Here we start to see some of the clumping of the stain. And these would be the chromosomes starting to become more visible. So the first stage of mitosis is characterized by the nuclear membrane is going to break down and the chromosomes become visible as clumps of stringy things. All right. Now at this stage, we've already suggested that the chromosomes have already been duplicated here. The chromosomes were duplicated during interphase of the cell cycle. So these chromosomes, when they first become visible in the first stage of mitosis, are already duplicated. And if we go back to the cell cycle again, remember we said that a cell with 10 chromosomes at some point is going to copy its chromosomes. That duplication process happens during interphase. Chromosomes are duplicated, and then at the start of mitosis, the chromosomes will become visible as those clumps, those colored bodies. So here we see the first stage of mitosis. We're going to call this the condensing stage of mitosis because the chromosomes are condensing. They're becoming visible. Here's some more chromosomes becoming visible. Here we see condensing stage, but now we also see in this picture, this cell over here has entered the second stage, has achieved the second stage of mitosis. We're going to call it the lineup stage because the chromosomes appear to be lined up along sort of the equator, the center of the cell. Again, remember they're duplicated here. We can think of the purpose of this lineup stage as the cell is sort of taking roll. So all chromosomes will be lined up here, and then the duplicated chromosomes are going to be separated to each side of the cell. So we have the condensing stage. The cells are visible as clumps, the chromosomes are visible as clumps, but they're not lined up. The next stage of mitosis is the lineup stage. Here we see some more pictures of cells in the lineup stage. Here we go here. This would be interphase here, right? Lineup stage. Another one, lineup. Nice one there, lineup. And now we're going to proceed to the third stage of mitosis, which we'll call the separation stage of mitosis. The duplicated chromosomes are going to migrate to opposite sides of the cell. Now, what we can't see in the plant is that there's going to be a set of proteins, microtubules and other proteins, in fact, that are going to organize and carry out this process of chromosome separation. But here we see lineup stage, and the third stage of mitosis is the separation stage of mitosis. Why separation? Because the duplicated chromosomes are separating to each side of the cell. Here we see some more cells separating, or rather the chromosomes are separating. We can sort of define maybe early separation, the chromosomes aren't too far apart. Late separation, the chromosomes are farther apart in the cell. And now we get to the final stage of mitosis, the division stage. So the chromosomes are real far apart in this cell here. In fact, they're kind of bundling up. And in time, a nuclear membrane is going to reform around these bundles of chromosome. But then what's going to happen is the cell is going to split into 2 cells. Now a new membrane is going to be constructed here, and in the case of plants, a cell wall, cellulose. Right, the cell wall will be constructed here, forming 2 cells. Some more pictures of the last stage. You can almost see the cell wall a little bit there. So at this stage then, when the cells have divided here, we have now two new daughter cells. The nuclear membrane will reassemble around the packages of chromosomes, and the cell will enter interphase again. So in that final stage, there is a difference between plant and animal cells. For in plant cells, there is the cell wall that's constructed. In addition to the cell membrane, a cell wall has to be built. In animals, the cells only pinch in their membranes to form the daughter cells. So mitosis then can be summarized in these four stages. Condensing stage, the nuclear membrane breaks down, the chromosomes become visible as those stringy structures. They are condensing. The lineup stage, the duplicated chromosomes are going to line up. And then the separation stage, the duplicated chromosomes are going to separate to opposite sides of the cell. And then the division stage, the actual cell will divide into two daughter cells. And if we think about what's the point of this whole process, the whole function of mitosis is to ensure that a copy of hereditary information is present in each daughter cell. So the whole dance of the chromosomes here is to ensure that daughter cells receive a full set of chromosomes, which are the hereditary molecules. And they contain the full set of recipes to make all the different proteins in the cell. One last look at this kind of picture again. So now, looking at that same picture, we can identify some cells in different stages of the cell cycle. Remember, if you don't see any chromosomes, you're just an evenly stained nucleus, you're in interphase. That is not a stage of mitosis. It is that other much longer stage of the cell cycle. Here we see a lineup cell, and we've got 3 cells here, maybe 4, in some stage of the first stage of mitosis, condensing. The DNA the chromosomes are kind of coiling up, what we call condensing. And so the stain takes on the shape of these little squiggles. Those are the chromosomes becoming visible.